The overall goal of the following experiment is to monitor ligand-receptor interactions with fluorescence resonance energy transfer. The method presented here utilizes an interesting property of PDA. Blue shift in the UV-Vis electronic absorption spectrum of PDA after an analyte interacts with receptors attached to PDA. First, polymer diacetylene, or PDA, is synthesized, and PDA liposomes are prepared. Next, rhodamine-tagged BSA is bound to the PDA liposome surface. When fluorescence resonance energy transfer is then measured, the emission spectrum of liposome-bound BSA rhodamine overlaps with the absorption spectrum of PDA. This satisfies the resonance requirement for the FRET mechanism. Thus, data can be obtained that indicate interactions between ligands and receptors.
The main advantage of this technique over the existing methods like ELISA is this method is very simple and inexpensive. Since fluorescence is intrinsically more sensitive than colorimetry, we can have the detection limit at very low sub-nanomolar or lower concentrations. The implication of this technique extends toward bacterial and viral sensing because it can selectively sense those microbes at very, very low concentrations.
We first had the idea of this method when we found the effectiveness of FRET-based polydiacetylene sensors in 2010.
Let's say we have two molecules, donor and acceptor, and both molecules